Welcome to my YouTube channel BioBits by RK. Ever wondered why doctors quickly classify bacteria as gram-positive or gram-negative? This classification can decide the right antibiotic to fight infections. Let's unravel the mystery behind gram staining. Gram staining, developed by Hans Christian Gram in 1884, is a fundamental technique in microbiology. It helps identify bacteria based on their cell wall structure, crucial for diagnostics and research. Let's break down the gram staining process into 4 key steps. First, we apply crystal violet, which penetrates all bacterial cells. Next, iodine acts as a mordant, binding the stain firmly to the bacterial cell wall. Then, alcohol is added to decolorize. Gram-positive bacteria retain the violet color, while gram-negative lose it. Finally, safranin is added. Gram-negative bacteria absorb this red dye, while gram-positive remain purple. But why does this happen? It's all about the cell wall. Gram-positive bacteria have a thick peptidoglycan layer, which traps the crystal violet. Gram-negative bacteria have a thinner wall and an outer membrane, so they lose the initial stain. Gram staining is more than just a lab procedure. It's a life-saving diagnostic tool. Understanding this simple technique opens the door to advanced microbiology. Thanks for watching. Keep exploring the world of microbes. For more such upcoming videos, don't forget to like and subscribe to my YouTube channel and comment below on what topic you need more videos.